Hi, we're here to talk to you about the Calvin cycle. But before we begin, let's talk about where it comes from. You see, the Calvin cycle is a part of photosynthesis, which is actually a two-part system. Who knew? Crazy, right? The Calvin cycle is the second step, using ATP and ADPH that come from the first step called the light reaction stage. Well, that's all, folks. Enjoy, settle back, and get ready for a roller coaster of knowledge.
What is this?
Wow, Santa's really cutting back on the good gifts this year.
Why? I'd have to disagree, my friend. You see, that is only the gift that keeps on giving year after year. It's the thing that you're most grateful for out of anything. It's called the Calvin cycle. It brings tears to my eyes just thinking about it. Who are you? Are you ready to go on one of the most greatest adventures you'll ever know in your life? Things that you'll tell your children, and you'll tell your children's children, and tell your children's babies. What? Come with me, my friend, and I will take you to one of the greatest people that ever lived and died, because we're going back in time.
Eureka! I figured it out. The Calvin cycle. Who are you people?
I don't know. Do you know where we are? No. Only the most amazing place in the world, in the planet of existence. Oh, no. Mr. Calvin, would you care to explain to this poor, ignorant, selfish child what you have just discovered here?
Well, I'd be happy to. Sit down. Have a seat. Well, the Calvin cycle is made up of 3 parts: carbon fixation, reduction, and regeneration of ADP. The first step is an enzyme, Ribisco, takes a molecule of CO2 and attaches it to a 5-carbon sugar molecule, which creates a 6-carbon sugar molecule. And then this 6-carbon sugar molecule is so unstable that it just breaks apart into 2 molecules, each having 3 carbon molecules. Move on to the second step, reduction, where ATP and NADPH transform these 2 molecules into G3P. G3P, what is that?
I don't know.
Glyceraldehyde 3-phosphate, which is used to make glucose. G3P, but only
Some are used to make glucose and the rest
to be used to make more BP, so then it can continue to repeat the cycle. Assistant, explain.
My name is Oliver, and I will be your assistant in guiding you through the Calvin cycle. The first step is carbon fixation. An enzyme named Ribisco will generate an attachment of CO2 to a 5-carbon sugar, resulting in a 6-carbon molecule that is unstable and will split into 2 molecules of a 3-carbon compound. Step number 2 is reduction. ATP and NADPH are used to convert 3-PGA into molecules of a 3-carbon sugar. NADPH will then donate electrons and reduce 3 carbons to make
G3P.
Now stay with me, folks. The third and last step is regeneration. Some G3P molecules will go on to make glucose, while others get recycled to regenerate the Ruby P acceptor. That's it, folks. I hope you enjoyed.
We're back! Yes, we are. So what'd you think of our amazing trip? Oh my gosh, it was like so great educational. Like, I love my gift now. Santa doesn't suck. Amazing, random lady. Thank you. You're welcome. Well, now that my time here is over, it's time for me to go.
Oh, thank goodness.